Hello, and welcome to the demonstration of ProteinWeaver, an intuitive web tool to visualize protein interaction networks in the context of their associated biological functions. Molecular interaction networks are critical for understanding protein function in interactions. While many network-based tools exist to predict protein function, existing tools do not specifically address where a protein is situated within the biological context. Current tools often have a steep learning curve, lack intuitiveness, and make it convoluted to reproduce results. In addition, these tools often focus on human protein-protein interactions, highlighting a lack of resources for non-human model organisms. To address these problems, we present ProteinWeaver. This web tool leverages gene ontology to answer the question: how does a protein of interest connect to other proteins with a specific function? Suppose you are studying flies and are interested in a protein like EGFR, and you want to know how it is related to a pathway like Wnt signaling. You can explore the relationship between EGFR and Wnt signaling using a protein interaction network where the nodes are proteins. ProteinWeaver quickly generates a protein interaction network with EGFR as the central protein, also known as the source node. Here we are showing a small number of the 233,000 protein-protein interactions in the fly interactome. A brief description of the query and Go term of interest is provided in the sidebar before selecting a node. If you want to learn more about the gene ontology of the query protein and Go term, links are provided to the gene ontology database entries in the sidebar. When you click on a protein, links to the database entries for the selected protein are provided in the sidebar. In this example, the square nodes represent what ProteinWeaver calls Go proteins, proteins that are connected to the Go term and protein of interest. In this case, these proteins are involved in the Wnt signaling pathway, and these small circular nodes represent proteins on the shortest path from the Wnt signaling proteins to the source node, EGFR. Clicking on one of the Go proteins provides information about the ontology qualifier, how the role of the protein fits into the Go term of interest. ProteinWeaver uses a Yen's k-shortest path algorithm to generate the subnetworks. Users can adjust the number of paths to show if they want to increase the subnetwork size. A new subnetwork with more interactions is generated. ProteinWeaver keeps track of your journey, making it easy to revisit your queries, export data in JSON format, and export the current view of your graph as a PNG. This makes it an ideal companion for your research and collaboration efforts. As you explore the networks, ProteinWeaver makes it easy for the user to set any of the other proteins in the subnetwork as the source node. A new subnetwork is generated, making it easy and intuitive for users to explore further interactions associated with their Go term of interest. In conclusion, ProteinWeaver is designed for students, researchers, biologists, and anyone interested in understanding the world of protein interactions and their connections to biological functions. Currently, ProteinWeaver supports 3 non-human model organisms: Bacillus subtilis, Drosophila melanogaster, and Danio aereo. Work is underway to add more data for these species and support regulatory networks and interactions. In addition, to enhance the ability of users to explore networks through the lens of gene ontology, we plan to add the ability for users to move around networks through the gene ontology hierarchy. For example, increasing or decreasing the level of specificity of the Go term of interest. In addition, we plan to provide statistics about the total number of Go proteins connected to the protein of interest. We will collaborate with biologists working with the model organisms hosted on the database to explore networks and identify new and interesting relationships. Finally, I would like to thank the NSF for funding this work, and Larry Zheng and Derek Applewhite for their help as well. Thank you for your interest in exploring protein interaction networks with ProteinWeaver.